Hey everyone, and welcome. Every now and then you come across a scientific paper that reads less like a dry report and more like a full-blown detective story. And that's exactly what we're diving into today. We're going to uncover some incredible microscopic secrets hidden away inside some of Australia's tiniest creatures found on its most famous animals.
So let's just start with this idea. What if the biggest secrets of Australia's amazing wildlife weren't hiding in plain sight, but in its smallest, most overlooked parasites? It's a wild thought, right? To understand the big stuff, sometimes you have to look at the really, really small stuff.
All right, every good detective story needs a scene and some suspects, so let's get right into it.
So, the crime scene, if you will, is Victoria, Australia. And our subjects, well, they're some of the most iconic animals you can think of: koalas, wombats, and echidnas. But the scientists in our story, they weren't just there to admire these awesome creatures. Oh no, they were on the hunt for clues.
And here they are, our primary suspects, a total of 60 ticks. You've got Ixodes tasmanae, which came from 12 different koalas. Then there's Bothriochroton originans, found on a wombat. And finally, Bothriochroton concolor, which was collected from 2 echidnas. Each and every one of these tiny ticks is like a little witness, potentially holding some major secrets.
And that brings us right to the heart of the mystery. What microscopic life are these ticks secretly carrying around? Are we talking about harmless little passengers? Or something else entirely? That's what our DNA detectives were determined to find out. Okay, so with the suspects collected, the real investigation could begin back at the lab. Let's dive into how these scientists use modern tech to hunt for clues that are totally invisible. The method was pretty straightforward, really. Think of it like a 3-step investigation. Step 1, collect the evidence. That's our 60 ticks. Step 2, extract the DNA. So in the lab, they basically processed each tick to get all the genetic material out. And step 3, scan for bacteria. They used a technique called PCR, which is kind of like a genetic search engine, to look for the fingerprints of 3 very specific families of bacteria.
And wow, did that DNA dragnet pull in some surprises. So it's time. Let's bring in the microbial suspects for the lineup.
First up, let's look at the ticks they found on that single wombat. What was hiding inside them? Well, it turned out to be one of the biggest shockers of the whole study. Get this. They found a dual infection. These ticks weren't just carrying one type of bacteria, they were carrying 2 different kinds at the same time. One was a Rickettsia-like bacteria, closely related to a known human pathogen, and the other, a Coxiella-like bacteria, which is a close cousin to the nasty bug that causes Q fever in people. So yeah, not exactly harmless passengers. And this wasn't just a one-off. A whopping 70% of the ticks from that wombat were carrying both of these bacteria. That's not just a coincidence, that's a pattern, a really significant one.
All right, next in the lineup, the ticks from the echidnas. Now, echidnas themselves are these ancient, incredible creatures, right? So it's kind of fitting that their ticks were hiding a secret that was just as ancient. This was a huge deal. They found a totally unique bacterium. Its closest genetic match was something called Rickettsia bellii, which is part of a really old ancestral group of these bacteria. And here's the kicker. This was the first time ever that this ancient group of Rickettsia had been recorded anywhere in Australia. A true landmark discovery.
And that brings us to our final suspects, the koala ticks. Remember, this was the biggest group, collected from 12 different koalas. And, you guessed it, they had their own unique story to tell. So, in these ticks, they didn't find Rickettsia. Instead, they found something related called Rickettsiella. You can think of it as a cousin, same family, different branch. But what they found was wild. They identified 3 different types. And check out their closest relatives. One type was related to a pathogen you find in crickets, another was related to one in crustaceans, like pill bugs, and the third was related to a type known to infect mammals. I mean, talk about unexpected. That's just an insane amount of diversity hiding in one species of tick.
So after all that, what's the final verdict? Well, like any really great investigation, this one didn't just close the case. It blew it wide open with a whole bunch of new mysteries.
I think the researchers who did this study put it perfectly. They said their findings point to, and I'm quoting here, a previously unrecognized high degree of bacterial diversity. Basically, it's like they peeked through a keyhole and found a whole new universe on the other side that no one even knew was there.
So, The big takeaway here isn't about solving a single mystery. It's about creating a brand new map, a map to a hidden world of microbes that's been right under our noses this whole time. And like any new map of an unexplored territory, it raises way more questions than it answers.
And the questions are huge, like, are these just new strains of bacteria we already know about? Or are they completely new species? Are they dangerous to the koalas and wombats? And the big one, of course, could any of these jump to humans? What are they even doing out there? What role do they play?
And that's really why all this matters. This isn't just some fun, microscopic whodunit. Understanding these hidden ecosystems inside parasites like ticks is absolutely crucial. It's how we keep an eye on the health of wildlife, and maybe, just maybe, stay one step ahead of the next new disease that could impact us.
So, the door to this hidden world has just been cracked open. The investigation is really just beginning, and you have to wonder, now that we have the map, what other secrets is it going to show us?